So just like Ali mentioned, it'd be really cool if this was somewhat interactive. So maybe asking you all for responses at various points. Please do not be shy and respond the best you can. So today I'm going to talk about a few aspects of early vision. I'm going to give an overview of computations of early vision. This will stem from just a really brief description and overly broad description of anatomy of the retina. I'll also talk about receptive fields and how the visual system has a key difference between the kinds of computer vision systems that we all use. Just to anticipate a little bit, we use convolutional operations, whereas the visual system replicates the machinery needed to process information across the visual field. I'll also talk about neural tuning curves, which is an extension of these neural computations, or what I will talk about with neural computations, and moving us into cortex. And finally, I'll take a more perceptual perspective on early vision and talk about how spatial resolution, spatial frequency interacts with our understanding of the world. So let's start with a super simple diagram. Obviously, this is like from high school biology. Diagram of the brain and our view of the world. So hopefully all of us have two eyes. Those two eyes take in views or images of the world that are not entirely overlapping. So information from the left eye is going to project ultimately to the right side of cortex. Information from the right eye is going to ultimately project to the left side of cortex. So in a moment, we're going to talk about some of the initial machinery responsible for this transduction of visual information. And I'm going to skip over an intermediate, although very important stage of processing implemented in the lateral geniculate nucleus. This is a part of the thalamus, but it's not really relevant for this really broad overview today. And we'll jump right into early visual cortex, also known as, I guess it's cut off here, but the striate cortex. This is also known as V1, so excuse me if I jump between those terms. I'll try to be consistent. All right, so as Ali mentioned, the eye itself is incredibly complex in terms of its architecture, and the whole purpose of this complexity is to take image reflecting from the world and focus it onto the retina at the back of the eye. And if we zoom in, take electron microscopy image of the retina, we can see the cones that Ali mentioned. So here's just the bumps are cones in this image here, and you can see bumps of different sizes. And this is because different cones are sensitive to different wavelength spectra of light. So you have it's very faint, but the arrows there are pointing to these B cones, so blue spectra. So already we can start to talk about the computations in early vision, even at this level of the retina, which to me is kind of shocking and very cool. So Ali provided a much more intricate overview of that anatomy, but let's just boil that down into three distinct stages. First, there's the photoreceptors, the cones that are going to take in light reflecting from the world. There's these intermediate bipolar cells, horizontal cells, amacrine network, all that stuff is also there, but we're going to ignore that for now. We'll just say there's these intermediate bipolar cells, and these bipolar cells project onto retinal ganglion cells. So what we know then is that if you stick an electrode in a retinal ganglion cell, an RGC, you're going to record signal of those cells as they respond to the world around us. And if you start to flash different kinds of images, stimuli, if you change the world at all, you'll see that a cell may respond to some things, but not to others. And the way these cells respond is through a currency called the action potential. So I'm sure you all have heard this, but it's an electrical impulse emitted by a cell down something called the axon. So this is the output of the cell. So these happen very quickly. This is an all or nothing process. And essentially, the pattern of these action potentials over short bins of time are what's important. So this is a little bit up in the air for different cells, different parts of the brain, different kinds of computations. What is the critical period of time for action potential communication? But you can roughly think that summing up the number of action potentials, just taking some rate computation summary of the action potentials over a brief period of time can give you an idea of what a cell is doing. So now we have this network which goes from cones through bipolar cells to the RGCs, and we can cast that as a computation. So one way to think of this is that the cones are something like X's or input images, let's say. The bipolar cells and their projections onto RGCs are something like weights. And then the RGC itself is integrating the weighted signal that is passed through these bipolar cells. And as I mentioned, there's action potentials on the output, so we have some nonlinearity on the output of the RGC, so with the function f there. And really what this is doing, there we go, really what this is doing is simply some rudimentary form of template matching. So there's going to be some preferred stimulus, and the evaluation here is how similar is the stimulus falling on or being projected to the cell to its preference. Up in the right-hand corner, you can see just one possible activation function this can take. Of course, as we all know, you can pretty much put anything on there, and at least with backprop, it'll work. In the brain, it's something closer to this kind of half-wave rectification, which I'm not even depicting here. And so, let me go back. As I mentioned, this concept of template matching is very powerful, and it's also a pretty accurate description of what's going on in early visual processing. So this is some seminal work from David Hubel and Torsten Wiesel. If you haven't heard of them, they won the Nobel Prize for physiology based off of their discovery or mapping out of all the processes in early vision. And this was all done in cat visual cortex. And so what they did is they anesthetized cats and stuck electrodes into various regions of the cat cortex, or retina. So here you're looking at responses in these retinal ganglion cells to different kinds of stimuli. So the responses are the hatch marks recorded across time. And you can see the record when the stimulus is on or off. And examples of the kinds of stimuli that elicited these action potentials are shown with the circles. So at the top is a so-called off stimulus, so nothing is on screen. And if the stimulus was responding to stuff that is there, if the RGC was responding to stuff that is there, then it should not fire when there is nothing on the screen, just to illustrate that logic. Below that is the circle, the white circle, which is a center on off surround stimulus. Below that is this bigger circle, which covers both a small center responsive patch of a neuron, as well as the surround. And below that is this donut-like stimulus, and this is something like if a neuron or an RGC will respond to the presence of stuff in the surround, but not in the center of a small patch of an image. And so what I'm showing you here is on the left is the response profile of an on-center off-surround cell, and on the right is an off-center on-surround cell. And so RGCs can, for the most part, just be binned into these two categories of response properties. You're going to be either on-center off-surround or off-center on-surround. So what does this really mean? Well, in the top right-hand corner, you can see a cartoon of what this might look like. Here below that is a template of that kind of sensitivity, which as computer vision experts, you can imagine just convolving with an image. And the output of that convolution would be something like what I'm showing you in the bottom left-hand corner, this neural image, or an image, a processed version of the viewed image in which the edges are emphasized. And so this template matching, again, I keep coming back to it, but it's so powerful and so prevalent throughout biology. And just as a brief aside, I want to introduce the jumping spider. So this spider has 4 pairs of eyes. It is also used as a model for learning and for visual learning because of its great visual acuity. And one thing we can take from the jumping spider is that when probing its rudimentary vision system, you can see very strong selectivity for relatively complex kinds of stimuli. So in the top left-hand corner, you can see different kinds of stimuli that go from not resembling a spider to more so resembling a spider. And below each stimulus is the response of the probed part of the spider's brain. And so what you can see is that as the stimulus starts to look more and more like a spider, this probed part of the spider's brain responds more. And this is surely surprising because these spiders, again, have such rudimentary vision systems, but that this means this template is encoded through some either genetic or rapid learning procedure that the spider undergoes. Similarly, you can look and see that there's templates for relatively complex shapes or flies that the spider also responds strongly to. Okay, so now that we know, yes? Yeah. No, it is not. So color sensitivity is thought to mostly emerge in V1, V2. So of course are there any retinal experts? I mean, color sensitivity in RGCs?
So you have RGCs that are attached to different cones. So you have 3 cones in the retina, red, green, and blue, in addition to rods, which are just light-sensitive detectors. And so you'll have RGCs that will create center surrounds in there that are based on individual cone types, and you won't tend to have much of a mix between the colors at that level. When
That's
you
right.
start getting red and green opposing in center and surround, by the time you get up to the LGN, and that complexity increases as you go along the visual system.
Cool, thank you. So moving on to this concept. So now we've talked about computations in early visual cortex. A dovetailing concept is that, as I mentioned, the computations that are performed are yoked to specific parts of the visual field. So this is called receptive fields of neurons. So for an example of this, imagine 2 RGCs, which can have different response properties. So one could be on-center, off-surround, while the other having the reverse response properties. Those 2 RGCs could nevertheless be overlapping in space. And this is desirable, of course, because then at every location in space, you can perform the full array of computations necessary for really understanding the world. And to give you an idea of how this is typically done, so this is work from Hupel and Weasel. I guess it'd be a lot more. Can you hear that? Yes, okay, good. This is work from Hupel and Weasel where they're trying to map out receptive fields for 2 different neurons, one that's on-center, off-surround, one that has the opposite response properties. And you can see that as they are, I guess it doesn't make sense with both movies playing at once, but you get the gist. As they're playing, showing the animal different kinds of stimuli, there's going to be action potentials firing in response to the stimulus shown in some locations but not others. And this, again, is to simply show that that we have receptive fields in cortex. And so what this does then is it gives us some interesting byproducts. One is the presence of illusions related to the locations or the asymmetries that these receptive fields yield. So I don't know if you can see this here, but there should be kind of greater intensities on the edges in this image, and that is thought to emerge from the presence of these center surround RGCs, or the increased activity of those RGCs on the edges in this image. There is no actual increased intensity at the pixel level. A similar kind of illusion, which is pretty cool, is called the Hermann grid illusion, in which this is scale dependent, so you may not be able to see it that well, but there should be some induction or change of the look at the gaps in this image. And so again, this is thought to emerge based off of, and this is pretty old work at the, right now, and there's more complex explanations for this now, but it's thought to emerge based off of just the location of these on-center, off-surround cells and how they interact with the stimulus. Another example of this, hopefully you can see that, is there should be increased intensities along the diagonals here. But of course, those intensities are not at the pixel level, but rather due to perception. So yeah, just to sum this all up, we have a full replication scheme of computation at all locations in the visual field. And one way that this was found is potentially morbid, but pretty cool. I believe this was a primate was put under anesthesia and injected. Its V1, early visual cortex, was injected with a dye that was taken up by cells that responded to a stimulus. The animal was shown the bullseye stimulus I'm showing you up at the top, which flickered. And so after sacrificing the animal and unfolding the cortical sheet, you could see that that stimulus was imprinted against the cortical sheet. So you really had this basically one-to-one representation of location in the real world on cortex. And what this also brings with it is something called cortical magnification. So you have a distortion in the cortical image versus how we perceive it, with parts of the periphery being magnified versus the fovea. And this is pretty cool because researchers have found that you can use this concept of cortical magnification to trick perception. So this is work by Jeremy Friedman and Eero Simoncelli, where they looked to previous research to map out the receptive field size, the area at which neurons in different parts of visual cortex respond to. So they looked in 3 parts of visual cortex. V1, V2, V4, that goes from early V1 to mid to high level visual cortex V4. And they found that the receptive fields, of course, expand as you go deeper into visual cortex. This is pretty much similar to the convolutional neural network receptive field size expanding down deeper layers of processing. And so what that allowed them to do then is create stimuli that tricked perception based off of this cortical magnification, which is, of course, with more distortion in the periphery than the fovea. And hopefully you can see that at the top. The technique involved something very similar to texture synthesis. This, of course, predated Matthias's texture synthesis work, but it involved within each of these predicted receptive field locations or cortical magnification locations, they performed a separate texture synthesis so that when you foveated on the red dot in either of those texts, you would have much more perturbation to the periphery than to the fovea. And so what you should be able to see is that you look at the red dot and everything looks fine, but then you fixate a little bit to the left or to the right and you see things are all screwed up. And you can do this in natural scenes as well, which is pretty cool. So on the left is an untouched original version of the scene, and C and D are versions of the scene in which different amounts of this cortical magnification perturbation was applied. And just closing the loop on the relationship between this stuff and texture synthesis, which is, of course, or style transfer, I should say, which is, of course, super popular now. So this work came from, I believe, the late 90s and early 2000s. And the difference between this and style transfer is that style transfer added an additional objective, which is to maintain some kind of content objective. Preserve the overall structure. They were less concerned about that here. They just wanted to use their cortical magnification predictions to perturb the image within those predefined spots. So now come with me to visual cortex, V1, and we can start talking about computations there. And it's old news by now. We can talk about the afferents, so the inputs to a neuron in V1, and those projections being W's, and the image that is passed through those projections being X's. And just as with RGCs, what neurons in V1, to a rough approximation, are doing is evaluating the dot product, the similarity between the image and their preferred stimulus. And so researchers have taken this concept and used it to evaluate what are called neural tuning curves in V1, or essentially the fall off in responsiveness of a neuron based off of changing stimulus characteristics. So this is work, again, by Peter Diane and Larry Abbott, where they probed a cell in V1 in early visual cortex and recorded its action potentials in response to stimuli. In the middle row, on the left-hand side, you can see the preferred stimulus, which is a oriented bar. And then as the bar starts to rotate, you can see that the action potential traces right next to it begin to diminish. And so if you plot the preferred orientation of this cell as a function, the responses of the cell as a function of its preferred orientation, you can see this bell-shaped tuning curve. And so these bell-shaped tuning curves are really ubiquitous throughout the brain when measuring responses and across different kinds of visual domains. So you not only find it with respect to orientation tuning, but also with respect to spatial frequency. So spatial frequency is illustrated at the top left-hand corner. I'm showing you different Gabor stimuli. Of course, along rows, those Gabors are changing in orientation. Across columns, they're changing in terms of their spatial frequency, which is just some unit in which you increase the number of the frequency of the stimulus. And so on the right-hand side, you can see responses to spatial frequency at a cell or different cells in visual cortex, and they also kind of have this bell-shaped tuning curve. This is more apparent when you probe for both orientation and spatial frequency at the same time. So in this study by de Valois in 1985, they found that there is a preferred, for a single neuron, you have a preferred orientation and a preferred spatial frequency. And when you think about these two concepts as orthogonal in space, you can get a pretty cool phenomenon, which will emerge from this video here. So building off of this idea of neural tuning curves, researchers have used techniques like what I'm showing you, which is called reverse correlation, to show a wide variety, kind of a black box approach, show a wide variety of different stimuli to an animal, to a subject, and record responses in neurons to those stimuli. So here you can see flashing are these on white bars and off black bars. And what you're hearing, hopefully, is action potentials elicited by the recorded neuron in response to these. And so what you'll slowly begin to see, I'll skip to the end in a second. Oh, there it is. On the left are, I believe, on responses for the neuron. On the middle are off responses. And if you overlay those two, what do you see? You see a Gabor-like function, right? So Gabors are indeed a very powerful way of modeling computation in early visual cortex. So now that we've talked about, just at the single cell level computation, let's extend out. How's my time, by the way? Great. Let's extend out to the population. So we know that you're going to need a lot more than a Gabor to understand the scene, and it turns out that this full replication scheme implemented in visual cortex is well prepared for that. So to illustrate this, on the left is a figure from David Hubel's work in which he inserted a probe obliquely through the cortical surface. Hopefully you can see that, but it's passing through just a few layers of visual cortex. So just to explain what that means, visual cortex is, of course, a cortex is a big sheet. And visual cortex in particular, if you probe through it perpendicularly to the surface, you'll see different layers of cell types. These cell types have completely different structures, and there's approximately, there are 6 different layers of cell types, but they're not really referred to as such, but just trust me on that. So there's 6 different layers of cell types, and what you're seeing in the yellow figure is a probe in red being inserted through a few of those at a single location in visual cortex. And so as that probe is being inserted, David Hubel was identifying the optimal stimulus for neurons that he was recording from. And on the x-axis is the distance that the probe is being inserted. On the y-axis is the preferred stimulus. And so you can see that the preferred stimulus changes as the probe gets inserted deeper. And this is really cool because at about 1.5 millimeters is when the probe changes receptive field. So really what you're looking at is basically full orientation tuning within a single receptive field, which is provided by a wide number of different neurons. So this is kind of a population code within a receptive field that can process any oriented stimulus at any position. And so this led to what's called the ice cube model, which I'm showing you on the right, in which you have these 1 by 3 millimeter chunks of visual cortex, which possess the full machinery necessary to process stuff in the world. That includes left eye dominance, right eye dominance, and binocular information. That includes color information and other properties that I'll show you in just a moment. But before that, I want to note that if you visualize this idea of changing orientations at any particular location in cortex, again, this doesn't mean receptive fields will only be responsive to one or another orientation, but rather just looking across cortex and visualizing the preferred orientation, you get this kind of pinwheel visualization where different colors correspond to different preferred orientations, which revolve around dots. So those are the pinwheels, those dots, and those are thought to correspond to these binocular sensitivity columns. So you can repeat this kind of game with other visual domains, with other stimulus properties, with spatial frequency, for instance. You can see that as you change the spatial frequency, you're going to see a similar kind of topography across cortex. Also with spatial tuning to oriented stimuli, oriented and moving stimuli, you're going to see a similar kind of response. And finally, if you visualize multiple of these response maps, topographies, at once, you can see that they are roughly orthogonal. So you're not going to have similar kind of overlapping ocular dominance columns and orientation maps, but rather these are going to be spread out in such a way that you're robust to these kinds of stimuli dimensions. Okay, so moving beyond neural tuning, let's talk about perception for a minute. So we talked about orientation tuning, and we talked about spatial frequency. We know, hopefully, from introspection, from looking at the world and looking at art, that these are 2 important properties for understanding our surroundings. So on the left, you can see kind of the effect of lower spatial frequencies on our perception versus on the right. This would be higher spatial frequencies, which emphasizes edges and contours. And so work by Blackmore and Campbell back in 1969 sought to understand what is the optimal band of spatial frequencies that our visual system is sensitive to. This used a combination of rudimentary neural recording and psychophysics to say that it's approximately between 4 to 6 cycles per degree of visual angle. And when you go below that, you lose sensitivity. When you go above that, you lose sensitivity. So in this sense, the visual system is something like a bandpass filter, where you're going to be very responsive to this specific band of spatial frequencies. Of course, this changes from person to person and based off of lighting conditions, but as I'll show you in a second, it's a pretty good first approximation. So to give you an intuition about what this means, imagine looking at a face as you're talking to somebody about a meter away. We can calculate the optimal spatial frequency that the face is processed at by doing spatial frequency analysis of this image. And so at about 60 cycles of this image, you see that the edges of the face are emphasized. And if you go lower or higher, it's a little bit harder to see the image. And so we can take this approach and do some really cool stuff with it. So this is work by Odelia, and earlier with Philip Shins, where they took images of, let's say, dogs and cats, and transformed one into its low spatial frequency components, and the other into its higher spatial frequency components, and then overlaid the two. In the middle, you can see the overlay of the two, which should look like the one response I've asked from you all. What does it look like? A what? A cat. Interesting. Yeah, so this is all a function of the distance at which you're viewing this image. So if you're closer, you should see this as a cat, and that's because the high spatial frequency information is emphasized there. If you're further away, you should see this as a dog. And we can do the same thing with gender. We can overlay two different gendered faces, and at different viewing distances, you should see one gender versus the other. We can do the same thing with famous people in history, with Albert Einstein and Marilyn Monroe. That's good. And work by, earlier work looking at Mona Lisa has suggested that her smile is in fact, or the perception of her smile is in fact a function of the spatial frequency that it's embedded in. So in lower spatial frequencies, you should see more of a smile than in higher spatial frequencies, meaning that if you're further away from her, you should see more of a smile. If you're closer up, you shouldn't. Or if you view her in your periphery, you should see more of a smile than if she's in your fovea. And so this whole idea of different spatial frequencies differently influencing vision has come to a head in work where Odelia and Philip Shins showed that at these two spatial frequency bands, we are sensitive to them at different times at different times of processing visual information. So if you do the same kind of trick where you take an image of a building and an image of a road and overlay the two, and flash the image very quickly, the task here is to answer whether that image is the same, whether the hybrid image is the same as the probe image shown below. Well, if you do this very quickly, you're going to see that people more often than not view the hybrid image, view the lower spatial frequency components of the hybrid image. Whereas if you speed it up, if you slow it down and give people more time to process, they're going to view the higher spatial frequency components. And so this is pretty cool because it yields this hypothesis that we're using these two bands of spatial frequency to process our environment in different ways, with an initial low pass or gist understanding of our environment to analyze the general layout of where we are, and subsequent foveations or saccades to important parts, allowing us to have more high resolution, high spatial frequency analysis and better understand our surroundings. And just to give you an idea of how effective this is, you can take images of scenes and you can perturb them in such a way to ruin any real perception of their components. And yet you can see that the image on the left, which is perturbed, it has a much wider expanse than the image on the right, and the image in the center has converging lines, unlike either of the other two images. More quantitative analyses of this kind of thing have shown that indeed this low pass filtering, this gist style analysis of images is a really good first approximation for seeing categories. So to today I've told you about neural computations in the RGCs, or in the retina, as well as in early visual cortex. I left out a huge amount of information, of course. This is just a brief overview, and I encourage you to look at other kinds of operations, including recurrence and feedback, and how those might influence processing in these areas, particularly in V1. I've also talked about receptive fields and how receptive fields are a key difference between how humans, how biology processes the visual world versus how our machine vision models process the visual world. I've talked about neural tuning curves and how in V1 we have the full replication of all the necessary machinery to process basically any kind of visual information at any receptive field location. And finally, how information at different spatial frequencies are used to inform our behavior and interactions with the world. So that's all I have. Any other questions? Yes? Yeah.
Is that sort of like, what would its optimal thing would be, like blue versus black, or is it blue versus another?
Oh, I see what you mean. Yeah, so the colors here, I didn't explain that, but it's a color code for orientation. Does that clarify? Yeah, I didn't explain. So color vision is a far more complex phenomenon, which Ali is going to talk about. But yeah, so this is purely for orientation, and just to anticipate, there is coding for color, but it's debated how complex it is in early visual cortex. Yes?
How do we form the cortical image after this kind of like edge or kind of
So yeah, to answer that, I would say that the cortical image is the result of processing on the retinal image by these kinds of edge detectors. So, think about it in terms of a CNN. You have your input image, and then you have a first stage of processing, and you look at the activities, right? So this is something like the cortical image that you have up here. The distinction is that, of course, the processing units are going to be at all locations, are going to have all sorts of biological idiosyncrasies associated with them, and also a ton of different mechanisms that I didn't talk about here. Yes?
So the latest result that you showed with regard to the recognition with different spatial frequencies,
Yeah.
when you show it for longer period of time, we perceive the higher frequencies.
Yeah, this, whoops. Yes, keep
Yeah,
going.
it's okay. Yeah, there we go. Yeah, this one.
Sorry, animations, yeah.
Yeah. How do you know that this is not a different processes of the ventral versus dorsal pathway?
Yeah, well, so the hypothesis underlying this is that you do have different nerve fibers basically underlying the rapid versus slower processing. You have these magno versus parvocellular pathways. So I don't, other than to say that we know where the kinds of response properties that are in visual cortex, and we know the anatomy of these different nerve fibers, and so it points to this being a ventral stream function. Dorsal stream, you would suspect that you have, of course, it's well known there's more motion selectivity, but then also if you do have any kind of orientation selectivity, that's going to be yoked to a specific motion direction. So, it's perceptual work, but I think we can be pretty confident that it's mostly a result of these different nerve tracts, and that this is a ventral stream process. Yeah, did you have a question? Can you clarify what you mean by reversible?
For instance, like a security
Yeah.
So I wanted to see in special transforming, and that is important for us to follow.
Yeah.
Do you know or are you aware of any work that or do you
Yeah.
have an idea how this is done?
I mean, so this would be a nonlinear function where you just like shut off some of the frequencies. So maybe I don't know of any work like that. It's an interesting question. If you can think of some differentiable way of doing this, where you can, get the inverse GANs. I'm not sure. I'm sorry, but that's a really cool question, especially because this, it does look like you can embed some really cool stuff in these kinds of hybrid images. So, yeah.
Do you know, is there any feedback from later stages in processing to this sort of?
Yeah. So this is like a whole nother story that is still completely up in the air. So you get feedback to V1, you get feedback to LGN, which I didn't mention. LGN, just to show you some of the complexity of the kinds of feedback that you have. LGN is something like a relay station, but the way it's going to relay is a function of the current state of the organism, function of the recent history. There's going to be a lot of feedback based off of just task that's going to influence how you're relaying information. As early as V1, you get feedback such that you can decode, just taking neural recordings and then applying a classifier to V1, you can decode all different sorts of sensory information, not just vision, but you can decode auditory information in V1, suggesting that, again, there's some sort of integrated, maybe predictive coding scheme, something involving integrating other senses into the way that you're processing the current visual image. But beyond feedback, feedback is something like top-down connections, top-down processing. There's also recurrence. So just at the layer of V1 between cells, you're going to have these center-surround interactions. So in the ice cube model, I got to get off stage, but in the ice cube model, I mentioned that you have all this machinery within a single receptive field, but stuff that's going on within a single receptive field is going to be influenced by the context of the information in all other receptive fields. So it's a fascinating question and a completely open research question that I think will make a big difference eventually in the kinds of computer vision models that we make. So, yeah.
Okay, so